So, good evening, everyone. Instead of having our class on site today, so I just have this recording of the topic on the structure and function of animal cells. So this is just to supplement what you have performed in the laboratory last meeting. Yeah. So for the content of this presentation, so we will be looking into the structure of animal cells. So before that, let's define some important uh, information regarding animal cells or cells in general. So all cells have a plasma membrane. So this is also called a cell membrane. So it separates your cells from the outside environment or from its external environment. Then all cells have a DNA, so all cells have a genetic material that carries information that is transmitted from parent to offspring. Then all cells also have a cytoplasm. So this is a fluid filled um, area in which your organelles, which are small parts of your cells, are embedded with. And you also your cytoplasm, so it also consists the name of the semi-fluid present in the cytoplasm is the cytosol. And again, organelles are found in the cytoplasm. Then all cells share one kind of organelle called the ribosomes. So whether it's a bacterial cells, a fungal cell, an animal cell, a plant cell, so all cells that are found in the planet all contain ribosomes. So ribosomes are important parts of the cell because they participate or they are mainly involved in the synthesis of proteins. So again, this is the plasma membrane, and you can actually see this in your fresh mounts of the cheek cell. So this is just a very thin layer that separates the inner that separates the inner contents of your cell from its external environment. So it's not just a physical barrier from the external environment. Cell membranes actually um perform a lot of important functions in the cell. So, for example, cell membranes or plasma membranes, they are the ones that filter out um, the the entry and exit of particles, materials in and outwards the cell. So, there are a lot of things involved in this particular process. So, we will go over them in this presentation. So first, let's define what plasma membrane is and what is it made of. So plasma membrane is made of a phospholipid bilayer. So this is um, generally a lipid molecule composed of a hydrophilic head, meaning water loving. It readily dissolves with what in water. And it also has a hydrophobic tail, meaning it's water fearing, a nonpolar tail, and it's a hydrophobic tail again. So aside from the phospholipid bilayer or phospholipid molecules that makes up phospholipid molecules that make up the bulk of the plasma membrane, there are also other important um, molecules that are present in the plasma membrane. You have your cholesterols, so these are also nonpolar molecules that are involved with different functions in the cell. One of which is to um sup- to provide structural support in your phospholipid bilayer. Then you also have this different proteins that can be used as channels, transporter proteins for the entry and exit of materials. Then you also have sugar molecules that are embedded in the surface of plasma membrane. So most of these um, molecules are important in the recognition and cell signaling processes of a cell. So this is just an overview as to what you can see in a plasma membrane. So again, you can actually see this sa inyong slides last meeting. So yeah, so these are just some parts of your plasma membrane as discussed before. So we won't go over this in detail because I think your teacher in the lecture will actually discuss this in thorough we will discuss this thoroughly in our lecture classes. So again, your plasma membrane, it doesn't only contain your phospholipid bilayer, it actually has a lot of other materials embedded in it or attached to it. So for example, protein molecules, there are a lot of protein molecules in the plasma membrane, so they can actually function in different ways. Some are needed for the transport of materials, while some proteins present in the plasma membrane participate in chemical reactions that generate certain products or that breaks down certain components or certain molecules. Yeah. Then, as I have said before, the surface of the plasma membrane or cell membrane is coated or there are a lot of sugar attachments or carbohydrate attachments there. So you call that as the glycocalyx. So the glycocalyx, the main function of the glycocalyx is to provide protection from mechanical and chemical damage. It can also be very important in cell recognition because the um the sugar profile present in the on the surface of your cells can actually be used by different types of it can be used through cell recognition processes and cell adhesion. So let's just um not go into details sa kanang aspect because I think that would be too much for what we had in our laboratory. So again, just remember that the plasma membrane doesn't only contain the phospholipid bilayer. It is actually a mixture of different molecules. So sometimes that's why scientists also refer to the plasma membrane as the fluid mosaic because it's a mosaic of different molecules. Yeah. So one important aspect of cell membrane is the transport of materials. However, we won't discuss this today because next the next activities that we will be doing is on membrane transport or passive cellular transport in the cell. So yeah, we will be discussing this more thoroughly next activity on your next activity. Yeah. So let's look at the different organelles that you can find in an animal cell. So an animal cell, as we have discussed before, is a eukaryotic type of cell, meaning it contains a nucleus and it also has membrane-bound organelles. So the nucleus is a very important part of an animal cell or any cells in general, eukaryotic cells in general, because it contains the genetic material, which is DNA. So your DNA contains information that is transmitted through this machinery of your cells in order to create proteins. So, a lot of our bodily functions, physiological processes are really governed by our DNA because most of the things that happen within our cells are performed by proteins. So, we will be uh, discussing about this in thorough in our next nga mga lectures. But just know that the nucleus is very important again because it contains the genetic material. And the genetic material contains important information as to how a cell can function, how certain parts, certain components within a cell can function, and so on. So your nucleus, it also has its own membrane. So you call that as the nuclear envelope. So it separates the nucleus from the cytoplasm. and it is actually continuous with the endoplasmic reticulum. So endoplasmic reticulum is also a, an organelle that is present in animal cells. So it's a series of membranes that originate from the nuclear envelope. So in here, the blue arrow, that's your nuclear envelope. So you also have these vaults. So actually, vaults are just are one of the most recent discovery regarding organelles and organelles of cells. So it's one of the recent discoveries. So yeah, the main purpose of vaults is to transport material in and out of the nucleus. So your vaults, they can actually go through pores that you can find in the nuclear envelope. So it has some functions when it comes to communication within the cell. And currently there's still scientists are still really figuring out what makes up vaults and what's their primary function within the cell. So that's a just an not not interesting fact regarding vaults. So it's one of the most recent discoveries. Then you also have nucleoli. So the nucleoli is a darkened portion that you can find within the nucleus of a cell. So it is comprised of RNA and proteins. So this is also where your ribosomes, the protein synthesizing organelles, are found. So it's very prominent in cells that are synthesizing large amounts of proteins. And you have your ribosomes. So this organelle is very important since again, your ribosomes are involved in the production of synthesis and production of protein or amino acid chains. So this is your ribosomes, and they're usually attached to the rough endoplasmic reticulum. So it's called a rough endoplasmic reticulum because when you look at it under a electron microscope, you can really see the numerous amount of ribosomes attached to it. So that's why it looks very rough. So yeah, that's why it's called endoplasmic reticulum. And because it's and because ribosomes are attached to the rough endoplasmic reticulum, so its main purpose is in the synthesis and production of protein molecules. On the other hand, you have the smooth endoplasmic reticulum. So again, it's smooth because it doesn't contain or it doesn't have any ribosomes attached to it. So it has a different purpose, different function compared to your rough endoplasmic reticulum, and it's mostly concerned with the synthesis of lipid molecules within the cell. Then you also have a Golgi apparatus. So the Golgi apparatus is just a series of kaning mga membranous sacs that are present in the cell. So the main purpose of the Golgi apparatus is to process and sort proteins for transport. So the proteins that are produced by the ribosomes, some of them go to the Golgi apparatus, and in this organelle, certain modifications of the proteins produced by your ribosomes are being done, and afterwards they are being transported to other parts of the cell, or they transported towards the plasma membrane where later on they can be exuded or yeah, they can be exuded out of the cell. So that's the main purpose of the Golgi apparatus. So it's where proteins are processed and sort for transport. So for example, if the protein product is something that is excreted or secreted by a cell, so after it um gets processed here in the Golgi apparatus, it's transported towards the plasma membrane in the form of vesicles. So vesicles are just these tiny structures which has a membrane wherein your um end product, your modified proteins are contained. So once they reach the plasma membrane, they join with the plasma membrane because they are made up of the same components, so it can be easily absorbed by the plasma membrane, and the product inside can be secreted out of the cell. So this is how a Golgi apparatus, yeah, looks like. So information from the nucleus of the cell um, signals the production of the creation of milk proteins. So yeah, afterwards, that signal goes into the kaning rough endoplasmic reticulum and then goes to the ribosomes where the milk proteins, amino acids are produced. So after they are produced, the milk amino acid goes through the series of membranes until it reach through your kaning Golgi apparatus where it is modified and then transported to the plasma membrane where it is secreted out as a form of the milk that we're familiar with. So this is usually how it goes in mga secretory nga cells. As you can see, the Golgi apparatus participates a lot in this particular process. So you also have another type of organelle, which is the lysosomes. So basically, lysosomes are um, vesicles present in your cell that contains hydrolytic materials, meaning these are substances that can um, lyse or break down chemical bonds. So lysosomes, they're actually present in the cell so that unused na mga wastes do not accumulate within the cell. And rather than accumulation, these wastes can be broken down into simple materials that can then be reused by our cell. And at the same time, lysosomes can also be used to um, lyse different external na contaminants that are that were able to pass through the plasma membrane. Then you have peroxisomes. So it's still this is similar to the lysosomes. It's just that it has a um different na function. So peroxisomes, so it contains um it contains this enzyme. 'Di ba ang sa lysosome, the enzymes present are hydrolytic enzymes. But for peroxisomes, what you have are oxidative enzymes. So these enzymes, they actually break down hydrogen peroxide. So hydrogen peroxide is agua oxinada if you're familiar with it. That are the hydrogen peroxide that is generated by normal metabolic processes of the cell. Because if hydrogen peroxide accumulates within the cell, it can cause trouble in its normal metabolic processes. So that's why in order to remove them, certain enzymes, oxidative enzymes present in the peroxisomes break down hydrogen peroxide into much more simpler forms that are not toxic to the cell like water and oxygen. Then the cytoskeleton, so this is the um, part of the cell that contains contains. So it's the part of the cell that gives it structure and integrity. So it gives it structural integrity. So you might think na cells are just cells, they're just a lump or mush nga mga kaning nga mga yeah mga they're like mush or there really no something supporting them, but actually the cells contain their own skeleton, so that's why you call it cytoskeleton, because same with our skeleton, human skeleton, these cellular components give structural integrity to the cell. So yeah, We will not go over the different types of cytoskeletal components because this is beyond our activity last time, but just know that cells, these colorful na mga um these colorful na structures that you can find in this diagram all refer to the cytoskeletal components. So there are big na cytoskeletal components, there are intermediate, and there are small cytoskeletal components, and they have their own function and they have their own